morning, everyone. I welcome you all to the virology class. I'm sure for many of you, this is the very first time we are having a contact. Though you've had a class on virology earlier before now, taken by Dr. Mrs. Oyibamiji. I will be talking about the general properties of viruses. We'll be talking about general properties of viruses. Unfortunately, we are having a virtual class. I will have been able to at least to interact with each one of you better. My name is Mr. Balogun. Balogun is my name. Balogun P.O. By the grace of God, I'll be talking about general properties of viruses. By now, I believe you have a knowledge of what viruses are, and therefore I wouldn't bother myself speaking much about viruses, the definition of viruses anymore, or the history of viruses. It has been taken earlier. So, we want to look at the general properties of viruses. What are those things that are characteristics of viruses generally? Viruses are the smallest infectious agents, ranging from about 20 nm to 300 nm in diameter. That's talking about nanometer, okay? So they have nucleic acid that is encased in a protein shell, which may be surrounded by lipid-containing membrane. The entire infectious unit is referred or is termed as a virion. Referred to as a virion. So what we're saying invariably is that viruses are very small. They are very, very small, smaller than bacteria, and as such, it requires an electron microscope to be able to view them. So you cannot see them with an ordinary light microscope. You can't see them with an ordinary light microscope. So viruses belong to an independent status because of several distinguishing features which set them apart from other familiar microbial agents. You know, we're talking about other microbial familiar agents. We're talking about bacteria, talking about algae, talking about the fungi, etc. So there are certain things that actually distinguish viruses from all other microbial agents. For example, they are acellular. When I mean acellular, I'm saying they are not referred to as a cell at all. They are not a cell. You can't refer to a virus as a cell. So it is acellular. They replicate by a self-assemblage process and not by binary fission, budding, etc. as associated with other microbes. So what we're saying is that for viruses, each part of their body are being produced or synthesized independently. And the moment these are being synthesized, they come together, they are being assembled as if you're in a car factory, where you have the engine being produced somewhere else. You have the tire being produced or being made somewhere else. You have the main body being designed by a different company, and you have even the seats being made in different company. So you bring them together by a process of self-assemblage in order to make them a brand new and individual particle. So another important thing is that number 3, they are obligate intracellular parasites that infect virtually all types of cells. When I mean all type of cell, I'm talking about plants, animals, and bacteria, etc. So they infect virtually all types of cells. So, meaning an obligate intracellular means that you cannot find them, elsewhere. They must be, inside the cell. They live in between cells. So they live. They are not, you can't find them separate from cell. So number 4 is that they contain either RNA or DNA as their genetic materials. They never contain both. So unlike other higher animals where you have the RNA and DNA present in their cells, but in the case of viruses, you cannot have both the RNA and the DNA present inside of them. So they can only have one. So number 4 point is that they lack an energy generating system, and so they do not demonstrate any significant metabolic activity. Your normal or usual animal cell, plant cell, you have what is called mitochondria present in the cell, which is the powerhouse of the cell. But in the case of viruses, they do not have any of these organelles inside of them. So they don't have it at all. And as such, they lack an energy generating system. They do not demonstrate any significant metabolic activity. So no metabolic activity occurs. Number 6 point is that they depend on their host biosynthetic machineries for survival. We're talking about biosynthetic machineries, biological processes that takes place in the cells in which a virus has successfully infected becomes the energy generating system. And so viruses do not carry out any metabolic activities. They depend on their host for metabolic activity. Number 6, they depend on their host biosynthetic machinery for survival. They depend on the host for survival, and viruses are specific in the type of cell and host they infect. What that means is you cannot have a virus, that is a particular cell specific that can infect the epithelial cell, having it again being able to infect another part of the body. So, the moment it is specific for one particular cell, you cannot have it infecting a different cell entirely. You cannot have it. So, another very important one quickly is that they show variation in size and shapes, in structure, in their genomic organization and expression, their strategies of replication and their transmission. So they show variation. They are so diverse. So these are things that we must understand about viruses before we progress further. So further to this, we need to quickly understand some terms, the basic terms as related to viral properties. Number one, we need to understand what we talk about: capsid, the nucleocapsid, the capsomere, the virion. We talk about envelope, and defective virus and subunits. So I'll take them one after the other. We want to talk about capsid. They are symmetric protein shell, or the coat that encloses the nucleic genome, the nucleic acid genome of any particular virus. Capsid is just, the symmetric protein is talking about the proteins. They are arranged, in an orderly manner in order to form the coat. It forms the coat that encloses the nucleic acid genome of a virus. So the nucleocapsid represents the capsid itself together with the enclosed nucleic acid. So it's talking about the capsid itself, the plus the nucleic acid together is referred to as a nucleocapsid. So the capsomere are morphologic units as seen in the electron microscope on the surface of virus particles. It represents, clusters of polypeptides, clusters of protein chains, which, when completely assembled, form the capsid. So we have the virion. Virion is a complete infective virus particle. So a completely assembled infective virus particle is called virion. Just like I mentioned earlier, viruses, comes to exist only after the different parts of its body has been synthesized individually, and they come together to form a virus. So that infective virus particle is referred to as virion. Now the envelope is referring to a lipid containing membrane that surrounds some virus particles, and it is acquired during viral maturation by the process of budding through a cellular membrane. As you go further, you will understand how envelope comes into being in those viruses that have them. So, defective virus, a virus particle that is functionally deficient in some aspect of its replication. So, they are referred to as defective virus. While subunits refer to a single folded viral polypeptide chain. A single folded viral polypeptide chain is referred to as the subunit. Now I'll be talking about the structure of viruses. Structure, I want to look at how do they look like, or how does each of the viruses looks like So most viruses are extremely adapted to their host organism. Extremely adapted to their host organism, meaning you can't find them outside their host organism, apart from them. So hence, virus structure varies greatly. So since we have, diverse kind of viruses, as well as, the different host organism, so we can understand that what it takes one virus to adapt to a particular cell will be different from what it will take another virus to adapt itself to another different cell entirely. Of course, each of these cells have their different properties, and each of the viruses also have their different properties. So if a particular virus is going to remain adapted to a particular cell, then some things are present in that cell which may not be found in the other cell. So there are several, general structural characteristics that all viruses share in common, even though they differ in the type of host that they infect, as well as the properties. So, for example, all viruses have a protein capsid that contain its genetic materials, talking about the nucleic acid now, either the RNA or the DNA. Capsid constructions varies. Capsid construction varies greatly among viruses, with most of them being specialized for a particular virus host organism. Some viruses mostly are the types that affect animal and man, with membranous envelopes surrounding their capsid. So when talking about envelopes in viruses, are mostly characteristics of animal viruses. So this allows the virus to penetrate host cell through a process called membrane fusion. So when you get to the viral replication, when you get to viral replication, you will understand how different viruses, how they replicate. So the virus genetic materials rest inside the capsid. It is located or situated inside the capsid. So this genetic material can either be DNA or RNA, as I said earlier, but they never contain both. So the type of genetic materials a virus contains is used in viral classification. It is very, very key, as it forms a basis for viral classification. In addition to the nucleocapsids, some viruses, mostly those that infect bacteria, have a tail region, which is often an elaborate protein structure that aids in the binding of the virus to the surface of a susceptible host cell and the eventual release of the viral genetic material into the host cell. So if you look at each of these structures, they have different roles they play in the organism, on the virus that actually possess them. So we want to look at how small or how big are viruses. So this is taking us to variations in size and shapes. We want to look at, so that even if I want to have an imagination of what a virus looks like, I should, in my mind, be able to, to think of how it looks like. Viruses come in an amazing variety of shapes and sizes. So interesting. They are very small and are measured in nanometer. if I should ask any one of you, okay, how small is a nanometer? How small is the size of a nanometer? So you're wondering, is the nanometer, what does it look like? So a nanometer is 1 billionth of a meter. 1 billionth of a meter. So that is 1 part of a billion. When you have like 1 billion, and you're looking at, okay, 1 over 1 billion. Of course, you know billion. Now, megaviruses, for example, mimiviruses, as well as the Pandora viruses, are some of the largest known viruses, which range from 1 to 2.5 MB. That's in brackets, we can have 1 MB is equal to 1 million BP. BP is talking about base pair of DNA. Base pair of DNA. So between 1 to 2.5 MB is talking about the size of such viruses. And of course, as we said earlier, 1 MB is equal to 1 million base pair of DNA. 1 million base pair of DNA. So the amount and arrangement of the proteins and nucleic acid of viruses determine their size and shapes. The amount and arrangement of proteins, the amount of proteins, the arrangement, the way in which the proteins are arranged, and the nucleic acid of the virus itself determine its size as well as the shape. So it will determine whether it's going to have the helical structure, the icosahedra, etc. So the nucleic acid and proteins of each class of viruses assemble themselves into a structure called nucleocapsid, which I had already mentioned earlier. So going further, some viruses have more than one layer of protein surrounding the nucleic acid. Others have a lipoprotein membrane called an envelope, as we mentioned earlier, which is derived from the membrane of the whole cell that surrounds the nucleocapsid core, penetrating the membrane. So don't forget, I mentioned earlier that envelope formation or presence is a common practice, or a common characteristics or a common properties of animal viruses. So you may, find out the different animal, the range of animal viruses that, that exist. So you can do that yourself. So animal viruses exhibit extreme variation in size and shape. The smallest animal viruses belong to the families Parvoviridae and Picornaviridae, which measures about 20 nanometer and 30 nanometer in diameter respectively. You can imagine how small they are. Viruses of these two families are icosahedron in shape and they contain nucleic acid with limited, genetic information. Viruses of the family Pox Viruses are about 250 to 400 nm in diameter, and they are either they either can have polygons of neither polygons nor filamentous. So pox viruses are structurally more complex, than simple bacteria. That's very good information that you need to know. Animal viruses that have rod shaped. Rod shape is talking about the helical structure, helical arrangement. Viruses that have rod shape nucleocapsid are those enclosed in an envelope. These viruses are found in the families the Paramyxoviridae, the Orthomyxoviridae, Coronaviridae, and Rhabdoviridae. Not all envelope viruses contain helical nucleocapsid. Of course, those of the families Herpesviridae, Retroviridae, and Tugaviridae have polygonal nucleocapsid. So it's very important for us to take note of that. We can quickly go further. Looking at these different symmetric arrangements. So the shape and size of any given virus is related to the size of the viral genome. The shape and the size of any given virus is a function of its genome and the nature of the capsid proteins that are present. Some viral genomes are so small that they code for only a few structural proteins. For example, viruses that exhibit helical capsid symmetry have only one type of capsid protein. Only one type. So they don't struggle to arrange themselves. You understand? And as a result of that, it's going to be very easy for it to be arranged because only one type of capsid protein. Whereas viruses with other types of symmetry may have several types of capsid protein, and yet all these are meant to be arranged within the nucleocapsid. So, it is the interactions of these proteins subunits, the interaction between them that actually determines the specific geometric patterns of the various capsid. I come again. It is the interaction of these proteins, talking about the different structural proteins that are present. So it is the arrangement of these proteins subunits that determines the specific geometric patterns of the virus. That is, whether it's going to be a helical form, it's going to appear icosahedra, it's going to be complex. All this, they are all it determines how the structure of a virus will look like. So based on this, now we identify the following symmetry arrangements are identified. We have helical capsid symmetry. We have the icosahedral capsid symmetry. We have complex or combined symmetry. We have all this. So we shall be looking at them one after the other. I'm so sure that when you have the lecture notes delivered to you, you will be able to see each of these structural arrangements, how they look like, and of course it will be interesting to you. So I'm going to take them one after the other. Helical symmetry. We're talking about helical arrangement. Of course, it's talking about the rod shape. This composed of a single type of capsomere stacked around a central axis to form a helical structure, which has a central tube. It is in the form of a tube, a rod shape. And this arrangement may result in rod shape or filamentous virion, that is making the virus to appear a rod shape. The identical protein subunit of helical capsids are arranged end to end. They are not coiling around each other, they are arranged end to end in the shape of a rod, with each turn of the rod possessing the same number of subunits. Now, the non-covalent bond are formed between the amino acids on adjacent subunits of the same turn of spiral, as well as between the adjacent turns of the spiral. Okay. Now, the vast number of the non-covalent bond gives the capsid tremendous structural stability. So it actually confers stability to the it confers stability, and which is similar to the hydrogen bonding between strands of DNA molecule. If you have seen a DNA molecule, you understand what we're explaining here. The hydrogen bonds that exist, holding them together. So this produces a filamentous appearance of the virus. A good example of a filamentous, naked plant virus with helical nucleocapsid is tobacco mosaic virus, TMV. And while an example of an envelope helical virus is influenza virus. So you need to take note of that. You can go further to study each of these mentioned viruses so that you can know better about them. On the notes, I have different structures of the different arrangements of the capsule and the capsid symmetry that we have. When you get the notes, you will see them. The next one we have is the Icosahedra capsid symmetry. These are spherically shaped nucleocapsid constructed according to an icosahedra. Icosahedra is a polygon with 20 triangular faces. So you can imagine how big and complex that can be. So capsomeres that are connected to 5 neighboring capsomeres are called pentamers, while those with 6 neighboring capsomeres are called hexamers. If you have done your chemistry well, you will understand in your fundamental organic chemistry we have pentane, hexane. You can understand that these pentamers, hexamers, they are coming from that very angle. So it is worthy of note that as the size of a virus increases, the number of capsomeres increases. For example, adenoviruses have been shown to have about 252 capsomeres. You can imagine. Arrangements of nucleic acid in the core of icosahedral viruses appeared to be one tightly coiled unit in some viruses, while in others it appeared to be coiled fragments. So you can understand the difference between the icosahedral capsid symmetry and that of the helical structural arrangement. So quickly, let me just go to the complex or combined symmetry. This group of viruses possess capsid that is neither purely helical nor purely icosahedral, and many of them, and they may possess extra structures such as protein tail or a complex outer wall. These viruses have symmetric construction that are complex or may be referred to as a combination of a helical and icosahedral symmetry. Examples of viruses in this category are Pox viruses. We have the Off virus, and we have the T. even, that's talking about T. even phages. For example, we have the coliphages, those phages that affect E. coli, called the coliphages. So the T. even phages especially possess a collar structure located at the base of its head, which connects to a tail assembly that is in the shape of a helical sheet surrounding an inner hollow core. And it is through this core that the DNA located in the head can be injected into the bacterium during infection process. So when you get the notes, I'm sure you will have the structure of a typical bacteriophage, and you will understand how these things you understand the head, the tail, the fibers also that are present. They are such a very complex symmetrical arrangements. Here I have a table that shows the list of some viruses by name. We have different type of viruses. We have about 103 of them that were listed here. So when you get your notes, I'm sure you'll be able to. So you need to understand the different, the property of each of these viruses. The property of each one of them. You need to understand them. So let's take a look at the viral genome quickly. When talking about viral genome, of course, we are talking about the nucleic acid, which can either be DNA or an RNA. The viral genome is the molecular blueprint for the building of an intact virion. That is, it contains the genetic information needed for the replication of an intact virion. So it may be DNA or RNA, which may be double-stranded or single-stranded, linear or circular. Viruses with RNA genomes are called riboviruses, and those with DNA genomes are called deoxyviruses. Just as the name implies, deoxyribonucleic acid, deoxyribonucleic acid, and the ribonucleic acid. So, plant viruses, for example, generally possess RNA genomes with a few exceptions, such as the cauliflower mosaic virus, CMV, which contain DNA. On the contrary, animal viruses and bacteriophages, on the other hand, generally possess DNA. Only very rarely they possess RNA. As it is true in all forms of life that nucleic acid of each virus encoded the genetic information for the synthesis of proteins, in almost all free living organisms, the genetic information is in the form of double-stranded DNA arranged along the length of a double helix molecule. Of course, the DNA has been established to be a double helix molecule. In viruses, however, genetic information comes in variety of forms. They come in variety of forms, and on the basis of this, the basis of the number of strands in the genome, 4 types of viruses are recognized. 4. We have the single-stranded DNA viruses. Single-stranded DNA viruses, an example of that, we have the coliphages. That's the viruses that infect E. coli. We have the double-stranded DNA viruses. Example of such, we have the herpes virus, the smallpox virus, Vaccinia virus, etc. We also have the single-stranded RNA viruses. An example is the polio virus and the TMV, tobacco mosaic virus. And lastly, we have the double-stranded RNA viruses, which is typified by the reoviruses. The reovirus, an example. That's what that means. In single-stranded RNA viruses, the genome may be plus-stranded RNA or a negative stranded RNA, so it can be positive or negative, and it is said to be plus when it functions as a messenger RNA. It functions as a messenger, or minus strand when it functions as a messenger RNA. So in some cases, Virion possess a segmented genome. Virion may possess a segmented genome, e.g., we find in automyxoviruses which are minor strand, it is a negative strand RNA molecule. So most double-stranded RNA viruses have segmented genomes, with each segment representing a single gene that encodes the significant information for the synthesis of a single protein. What we're saying is that each of these segments represents a gene and it encodes the information for the synthesis of that single protein. So the virion of most plant viruses, many animal viruses and bacterial viruses are composed of single-stranded RNA, and in most of these viruses the genomic RNA is termed positive strand. Like I said earlier, it's said to be positive because the genomic RNA acts as an mRNA, as a messenger RNA, for the direct synthesis or the translation of viral proteins. It serves as a direct, it serves as, because the genomic RNA acts as messenger RNA for the direct synthesis of the viral protein. So take note of that. Several large families of animal viruses, for example, the Rhabdoviridae, particularly contain genomic single-stranded RNA termed negative strand that is complementary to the mRNA. So what I'm explaining invariably is that when we say a particular genome is negative stranded, single-stranded RNA, we are saying it is negative when it is complementary to the messenger RNA. So all negative strand RNA viruses have an enzyme called an RNA-dependent RNA polymerase. They are transcriptase enzyme which must first catalyze the synthesis of complementary messenger RNA from the virion. That's from the virion genomic RNA. That is before viral protein synthesis can occur at all. So this variation in the nucleic acid of viruses is a very important factor in viral classification. Very important. Very, very important. Further to this, in single-stranded RNA viral family, for example, Retrovirudae, the RNA of these viruses is positive strand, but the viruses are equipped with an enzyme called a reverse transcriptase enzyme that copies the single-stranded RNA to form double-stranded DNA. In some viral family, they contain genomes that are partially double-stranded and partially single-stranded. An example of that is Hepatnav, of that family is Hepatnaviridae. So as I explained earlier, if you look at the Retroviridae, the retroviruses, an example, very good example of such a virus is the HIV. So, and that is why HIV is a very, very difficult virus, because of course there is going to be, there is going to be formation of it's going to be translated into a DNA eventually, which aligns itself on the host DNA, and as a result of that, it presents itself as part of the host genomic DNA. So I think I will end this here. While we take the next voice lecture from viral protein. Thank you. So now we want to look at viral proteins. Viral proteins. What are the functions of viral proteins? What do they do in viruses? We want to quickly look at them. Viral proteins. The proteins determine the antigenic characteristics of the virus or viruses generally. Hence, the host protective immune response are generally directed against antigenic determinants of proteins, or the glycoprotein exposed on the surface of the virus particle. It is important to know that it was earlier believed that viruses lack enzymes, but now some enzymes have been discovered. Some viruses have been discovered to contain enzymes. So some viruses carry enzymes. Enzymes, of course, are proteins that are present inside their virions. For example, the spikes of enveloped viruses like influenza, measles virus, and mumps contain the enzyme neuraminidase, which facilitates their penetration into the host cell. You can see that. In some cases, the spikes contain hemagglutinin that allows the clumping of red blood cells and help in adsorption to specific host cell. For example, it happens in polioviruses, adenoviruses, influenza, measles and mumps, etc. The tips of bacteriophage or bacteriophages contain the enzyme lysosome, which enhances penetration into the host cell. So each of these enzymes play one role or the other in viruses that contain them. In retroviruses such as HIV, the Rao sarcoma virus, and RNA-dependent DNA polymerase called reverse transcriptase found associated with its genome, which help in DNA synthesis from the viral RNA through the process called reverse transcription. So let's just look at, generally the structural proteins of viruses, their functions. One after the other. I want to look at the functions of structural proteins associated with viruses generally. Number one, they facilitate the transfer of viral nucleic acid from one host cell to the other. That is, it facilitates the transfer of the viral nucleic acid from one host cell. The moment an infection occurs, before the nucleic acid is being released, attachments, they attach themselves, of course, attachment on coating. When you get to viral replication, there are some of these things that will be explained to you, because for a successful infection to occur, there are processes that must take place before a successful infection of any cell, of any virus in a particular cell. So number two is that these proteins protect the viral genome against inactivation by nucleases. They protect the viral genome against inactivation. And number three, they participate in the attachment of the viral particles to a susceptible host cell. They participate in the attachment of the viral particles to a susceptible host cell. And number four, they provide the structural symmetry of the virus particles. So it is a function of all these proteins to provide structural symmetry. Protein provides structural symmetry because the arrangement of these proteins, structural proteins, of course, is one of the is one of the determinants of how the size and the shape of a particular virus will look like. So a number of different viruses contain lipid envelopes as part of their structure. The lipid is acquired when the viral nucleocapsid buds through a cellular membrane during maturation, and the process occurs where virus-specific proteins have been inserted into the host cell membrane. This is typical of many animal viruses. I think I've mentioned this earlier to you. Viral envelopes also do contain glycoproteins, which, in contrast to lipids in viral membrane, they are encoded, and so they are not derived from the host cell. So they are not derived from the host cell. And it is by means of which envelope viruses attach themselves to target cell by interacting with a cellular receptor on the host cell surface. So there's always an interaction between the proteins, the structural protein we're talking about, and the host cell before any successful infection can occur. Now, there are certain things that we must understand looking at the physical and chemical agents, how they affect viruses also. They are still part of their properties, the general properties of viruses. There is a great variation in the heat stability of different viruses. That is the ability to tolerate heat varies tremendously among viruses, and which can be attributed to the symmetric arrangement of the capsid proteins. Viruses are known to be affected by physical factors such as heat, salt, the pH, radiation, and also chemical agents like ether, detergent, formaldehyde, etc. For example, Icosahedra viruses tend to be stable as they lose little infectivity after several hours at 37 degrees Celsius, which means they are stable. We're talking about their stability now. However, enveloped viruses are much more heat labile. When we're talking about heat, the being heat labile means to be easily damaged by heat. So they can tolerate heat to a greater extent. So viral infectivity is generally destroyed by heat at temperature between 50 and 60 degrees Celsius within 30 minutes. Viral stability is a very important factor in vaccine production and administration. For example, when you have your polio vaccine must be stored at freezing temperature to preserve its potency. And that is why when you see those that are giving children vaccine, vaccinating children on the streets of Lagos and other parts of Nigeria, you will see them that they put their vaccine inside some mobile container that have ice packs in order to maintain it at a very low temperature. So viruses are usually stable between pH value of 5 and 9, but majority of viruses are destroyed by alkaline conditions. So you can understand that the pH of 5 and 9 is not too acidic and not too alkaline. So we can understand that this pH range favors the survivor, and they are stable at that temperature, at that pH. However, alkaline pH have been known to affect viruses. Of course, an alkaline pH, talking about pH, those alkaline, anything above 7. 7 is neutral, of course. Anything below, even 5 is an acidic pH, but it is not too acidic. And also 9 is also an alkaline pH, but not too alkaline. So any alkaline condition that will affect viruses will have to be above 9. above 9, above pH of 9. So quickly, we just need to talk about viral crystallization. That is the crystallization of viruses. Crystallization of viruses. There are things I need to cover within this very, very week that we are in from our curriculum. So we have crystallization of virus. We're talking about crystallization. What does crystallization means? What does it mean? Crystallization is the process of transformation of viral components into organized solid particles. Crystallization of biological macromolecule, including viral components such as protein, is used to study structural characteristics. Of course, we know that viruses generally can exist as chemicals when they are outside a living host. So therefore, they can be crystallized. They can be crystallized. So, direct visualization of viruses, of course, became possible after the evolution of electron microscope. And of course, in 1935, tobacco mosaic virus became the first virus to be crystallized, followed by the successful crystallization of poliovirus also in 1955. A virus crystal consists of thousands of viruses, and because of its purity, it is well suited for chemical studies. Of course, the very first person, we cannot talk about crystallization without referring to the man, Dr. Wendell Stanley, who achieved the first crystallization of viruses in 1935. However, he was not the first person to try to purify TMV. But the methods available those days were not adequate to produce the virus samples that is pure enough to work with. So Dr. Stanley succeeded when he employed precipitation with lead acetate combined with a novel centrifugal method. The crystallized virus he achieved was strong enough that 1 cm cube of a solution containing 1 part of its liquid will infect a tobacco plant. Now it tells us how pure it was. Now the crystallized virus was found to be 17 million times greater than that of the heaviest known protein. Thereafter, other scientists later proved that the virus was not only a protein, as Stanley proposed, but a nucleoprotein. Hence, Dr. Stanley crystallized a living matter in a characteristically non-living form. A living matter in a characteristically non-living form for the first time. And of course, this discovery was a basis of his Nobel Prize for Chemistry in 1946. So when we're talking about crystallization, we're looking at the ability to crystallize, to form in the form of crystal, in chemical form, the viruses. That is, of course, we know that when they exist in a living host, they become living. But outside the living host, they are chemical. So, since they can exist as chemical outside the body, the process of achieving this chemical outside the body is referred to as crystallization. Thank you for listening.